Development of mites or life cycle of mites. I am here to report to you about the life cycle of a mite. The first stage is the egg larvae, pre-larvae, proto-nymph, joto-nymph, trito-nymph, and the last one is the adult. First, egg is a that many parasitic mites lay their eggs at places where they are protected from different climatic hazards and their access to the next host ensured. In spider mites or the family Tetranychidae, the eggs may be suspended or tied with silken guy wires to the surfaces on which they are laid. The number of eggs laid by female ticks varies considerably. In case of spider mites, mites from 20 to 200 eggs per female are laid. The next stage is the pre-larva. Pre-larva is a non-feeding form that occurs in acari and some other arachnids. In some cases, pre-larva appears to be little more than a featureless sac without legs or mouthparts. Extreme inhibitions of this type is referred to as calyptostasis and is found in some endeostigmata, Oribatida, and certain higher prostigmata. In other acariform groups, the inactive pre-larva may possess 3 pairs of legs, recognizable mouthparts, and setae, although mouthparts development and leg segmentation may be inhibited. But some scientists have referred to incomplete calyptostasis as elatostasis. Its extreme form of elatostasis is found in families Nanarchistidae, Adamistidae, and Anistidae. The pre-larva molts to the larva. The next one is the larva or larvae. Larvae of Trombiculidae are known as chiggers, and larval characters in Trombiculidae are considered very useful in species determination. The oorstigma present in some acariform pre-larvae persist in the larval stage when fully fed. Larvae of Pterogosomatidae, Trombididae, and water mites enter into a resting stage commonly known as nymphochrysalis which later emerges as nymphal stage after rupturing the old cuticle. The larval stage of virtually all species of argacid ticks differs from conspecific nymphal and adult stages in that it attacks and feeds on vertebrate hosts for an extended period, rather than the shorter attachment period which is the months to a few hours of the nymph and adult. Because larval stages attach to the host for the extended period, this life stage is typically collected more commonly than conspecific post-larval stages, and many argacid species are well known only from the larval stages. The next one is the nymph. Holotyrida, Oribatida, Opiliocarida, and many endostigmata, prostigmata, and astigmata have active protodeo and tritonymphal stages. A maximum of 2 nymphal stages occur in mesostigmata, or the protonymphs and jotonymphs, while in some prostigmata or the most heterostigmatina, there may be no phi nymphal stage. Only one active nymphal instar, the jotonymph, occurs in some prostigmata, with the protonymphal and tritonymphal stages passing as calyptostatic forms within the skin of the preceding stages. The nymphs of ticks have 8 legs like the adult but have no genital opening. Nymphal stages must undergo a critical waiting period for a suitable host. After engorgement, the nymph drops from the host, molts, and become an adult. Hard ticks have only one nymphal instar, the nymph becoming an adult after molting. Soft ticks may have several nymphal instars. And the next stage is the proto-nymph. Proto-nymph is the first nymphal stage following the larva is known as proto-nymphal stage. This stage is usually free active instar that may or may not feed. It is usually found in and adapted to the environmental location where the other nymphal stadia will occur. Only one nymphal instar occurs in most of parasite go-9 prostigmata that develop within the skin of the preceding instar. But there are two sequestered calyptostasis, one between larva and active jotonymphal instar and the other between the jotonymph and adult, or let's just call it teleochrysalis. Next stage is the jotonymphs. The jotonymph is the second nymphal stage in the life cycle and may differ from the adult only in size, the patterns of sclerotization, and cetacean. Phoretic jotonymphs of Europodina and parasitic Gamacida do not feed. Jotonymphs of many astigmatina are heteromorphic or are completely different morphologically and in behavior from preceding and succeeding stages. Such heteromorphic nymphs are called hypophyte or dihypophodes, which occur sporadically. Hypophylact functional mouthparts are principally phoretic stages. This special phenotype and of a jotonym can survive but environmental influences much better than the normal form as in family Diciphagidae. In most cases, the jotonymphs molt to the adult stage. Next one is the tritonymphs. In most cases, quiescent jotonymph molts to the adult stage, but in some groups of mites, the third nymphal stage or the tritonymph develops. It is usually an active stage but may be parrot in some members of Astigmatina. Tritonymphal stage is absent in the mesostigmata and may be absent and represented by a pharid calyptostatic stage or the teleochrysalis in many prostigmata. This stage is retained in some Holotyrida. The last stage is the adults. Adult mites are comparatively larger in size and sexually mature. In most mites, the adult is easily recognized by the appearance of fully formed genital regions and often has distinctive sclerotized shields. Sometimes they have an ovipositor spermatophoric organs or secondary sperm transfer systems. In case of ticks, in addition to egg stage, only 3 instars are recognized in life cycle, such as the larva, nymph, and adult. There is only one nymphal instar in the oxidative life cycle, but varying number of nymphal stages may occur in the argacid tick life cycles. So that's the end of my slide. Thank you.